Hi everyone. I don't really know how to follow that and all of the other incredible talks today, but yeah, a bit of an update on my research project and what I'm doing now. So I'm now a research fellow at the University of Leeds, but I recently completed my PhD, so that's what I'll be sort of covering today and the new projects that has led into. So a bit of background about what we do. So we work on immunotherapies for Ewing Sarcoma and osteosarcoma now as well. And background to why we use immunotherapies is quite important. So with the immune system and cancer, what goes wrong? Because initially, our immune system is great at detecting cells which have changed to a tumour cell or a precursor of what could be a tumour cell. And initially, immune cells are great and they will kill cancer cells. After time, the tumour cells adapt and change so that they can hide from the immune system. And this sort of results in an equilibrium where you get immune cells that are destroying the tumour, but the tumour is growing still. And ultimately, this leads to an escape phase, where actually there's complete loss of immune control and you get tumour growth. So the big question is, how can we wake back up the immune system so that it can fight the cancer again? So we do this, doing this with oncolytic viruses. I think especially after the past couple of years, the word virus sounds quite scary, but they are all viruses. Some of them are modified, some of them are just innate viruses that naturally occur. And the side effects of all of them are kind of colds and flu, so they are viruses that would just cause colds and flu in people. So what they do is they selectively target cancer cells while leaving healthy cells unharmed. So when they replicate within the cancer cell, so that's the first way in which they work. But then this results in activation of the immune system, and particularly we're interested in these natural killer immune cells. And this then means that the immune system is able to fight the cancer. So you have this two-pronged approach to fighting cancer with one treatment. So we have investigated a range of viruses. They are all made to clinical grade. I've been evaluated in clinical trials for other cancers, but it's just important to emphasize all of our work is preclinical and just lab-based at the moment. So as I mentioned, we're interested in these natural killer cells, or NK cells. And this is because of all the cells of the immune system, they are very, very good at recognizing cells which are infected with viruses and also cancer cells. So the oncolytic virus basically tricks the immune cells into thinking that there's an infection, which activates the immune cells to attack the cancer. So I've got a video, and I don't actually know how that will be played. Can someone do that? Oh, there we go, I think so. So the green cell on the video is an Ewing Sarcoma cell, so this is an example of what I've done in the lab. And the grey cells are the natural killer cells, and it's just really so you can visualize the NK cells and how powerful they are in destroying tumour cells. If you could play that again, because I think we missed it. Thank you. So yeah, you can see at the start a nice round tumour cell, and then it's destroyed by the NK cells which have been activated. Having a video was a mistake, wasn't it? Anyway, I'll move on to the next, but you just have to trust me that it looks really cool. Oh, there we go. So, initially in my research project, I wanted to investigate these two different mechanisms by which oncolytic viruses are able to fight cancer. So the direct killing of the virus against the tumour cells, but then also its ability to activate immune cells to kill the tumour. So this revealed that a panel of different oncolytic viruses were able to kill Ewing sarcoma tumour cells, and this rhabdovirus was very effective. And also, we found that if we have immune cells treated with each of the viruses, they have a significantly increased ability to destroy the tumour cell. So it's brilliant. You know, we were like, this is great, it's working in this model. But then later work where we used patient-derived samples. So the cell lines that I used previously have been going in the lab for tens of years, and these are more recently from patient biopsies. And actually, there were completely different profiles in response to the virus. So we're still seeing some level of response, but in blue you can see the response of the patient-derived cultures, and in red you can see the established cell cultures, and they're completely different. So obviously this highlighted that further investigation was required because these models were so different to the cell lines we'd worked with previously. So all of this data from my PhD was put together into new grants, and it led to the funding of 2 new postdoctoral research projects, which start this year, one this year and one next year, to investigate oncolytic virus therapy for sarcoma. So there is one project which is continuing this project in Ewing Sarcoma. So Dr. Fiona Errington-Mayes and Professor Graham Cook are working with Professor Sue Birchall on this. And then also another project which is investigating oncolytic virus therapy for osteosarcoma and see what those models show. So the Ewing Sarcoma project will be carried out by Richard, who's joining us from Oxford University. And basically, he will just be going on to look at basically repeating all the experiments that have been done previously. We have those models and assays set up. We just need to look more closely at them with the patient-derived cultures. And my project, which I am working on now, which I started last month, is investigating oncolytic virus therapy in osteosarcoma in preclinical models. So just like with my previous project, we are looking at the direct killing effects of the viruses, their immune stimulatory properties. I won't try to say that word again. And also, we want to determine how different immune cell profiles in tumours might affect how that's associated with patient outcomes, and potentially how we can modify that using oncolytic virus therapy. So conclusion, what the take-home message I hope is from this, that PhD projects are really important. I think someone did a really good talk earlier that was about how the smaller projects lead into bigger projects, and ultimately that's how you get to clinical findings. So PhD research projects provide findings for larger research, but also it provides the next generation, hopefully, of bone cancer researchers. So Hannah and I both started our PhDs at the same time, and we are both staying, so that's good, and I think we intend to for as much of our career as we can. And the overreaching goal is to progress these findings to clinical trials eventually, but there is a lot of preclinical work, so lab-based work, that is required before you can ever consider that. And it's an essential step in identifying new treatments. So I'd just like to say thank you. My supervisors aren't here today, they were here yesterday, but Graham and Fiona are absolutely wonderful, and yeah, a big reason as to why I've stayed in the lab that I work in. Thank you so much to Sue and Liz, and so obviously they've worked really hard to generate the patient-derived cultures and the surgical teams involved. They were so important in highlighting the differences in models. And yes, just a big thank you to everyone here. It's so important to me to be part of this BCRT community. It's been a massive part of my sort of career development, and I've always loved science, but actually it feels different. Like working in bone cancer feels like the community spirit is incredible. And there's been so many amazing talks today, so thank you.